slowby was called?
Great, well thanks Eileen, thanks for the brave souls that hopefully all have four-wheel drives that are still here, so that's awesome. Don't let the title fool you, we are in fact talking about round goby here. This is a species that we've been monitoring in central New York. Eileen, I'm not
area.
Oh. Sorry, people are coming in anyway. We got it. Awesome. So this is a species that we've been studying in the Canal system in central New York for a little while as we think about their eastward expansion. This is a figure that some of you might have seen earlier in another talk, but essentially we're talking about round goby. This is a non-native benthic fish, ballast water transplant that brought them to North America around 1990. They're really successful invader. They can spawn repeatedly during a single warm weather season, and they've got a number of negative ecological effects associated with them. When they move into new areas, you see the native benthic fish, your darters, your sculpin, your log perch often are extirpated. They love to consume drussidid mussels, which is good, but it means in areas with contaminated sediments, they tend to pick up a high contaminant load and pass that on. They're prolific egg predators. John Farrell and Rod Getchell will tell you they're one of the most prolific carriers of viral hemorrhagic septicemia, and they're increasingly associated with these avian botulism bird kills that we're seeing more of in the Great Lakes. More relevant to our study area, goby were first detected in Oneida Lake in late 2013. Lars and the folks at Shackleton Point will tell you that they had become the most abundant benthic fish by 2015. So, I mean, when you take a step back and think about that, you're talking about the largest inland lake in New York, from first detected to most abundant benthic fish, just under 2 years. Pretty astonishing. Also, during that same period, you'll see an angler captured a specimen. This is about 40 kilometers east of Oneida Lake. This was in 2014. This sparked a lot of panicked conversation really about what the current distribution of goby was in central New York at that time and how quickly we might see them expanding eastward into new watersheds. So this is a freeze frame of the 2019 distribution, and you can see once goby are at Utica, they've got a relatively short and unobstructed downstream trip through the canal system in the Mohawk River to ultimately reach the Hudson, at which point they can go north into the Champlain Canal into Lake Champlain, and it's 21,000 square kilometer watershed that spans 2 states, and south into the Hudson River estuary. Recent studies on goby salinity tolerance suggest that they can tolerate probably between 20 and 25 ppt, and it looks like their actual peak growth is between 5 and 15 ppt. So this is the Piermont site. It's about 40 kilometers upstream from the battery, and you can see our salinities this is just some RICOS data from 2016, but you can see in that given year, we're looking at peak salinity of around 10. So this suggests that goby are going to be able to colonize almost the entire Hudson River estuary, at least in terms of their salinity tolerance, other habitat requirements notwithstanding. So objectives of our project, we wanted to figure out the best way to monitor the invasion front, right? Anyone can go catch some gobies when they're packed in at 10 fish per square meter, a little bit more challenging when you're talking about 90 miles of habitat that the vast majority of which doesn't have goby, and where you do have them, they may be in low density, so a little bit more challenging. We're using benthic trawling, minnow trapping, bag seining, and kind of the cool, sexy method that we're hoping was going to work for us, environmental DNA water samples. So we're doing those 4 gear types at 12 sites distributed along the canal, spring and summer sampling for 4 consecutive years. This is a quick look at our sites. So this is at Sylvan Beach. This is our positive control site because goby were present there before we started in 2016, spanning all the way east, and this is actually below the Troy Dam in the estuary on the Hudson River. I'll blast through the methods quick. Seining, we're using a 30-foot bag seine, doing 3 pulls per site. Minnow trapping, G-minnow traps deployed in pairs, cheese and dog food, overnight soak. The trawling, we're using a Siamese trawl. This is similar to a mini Missouri trawl, 3 pulls at each site. And every year, I think this is the last time I'm going to use the next slide, and every year we have more traumatic experiences using this thing. The trawl is the method that we love to hate. We wish it would fail so we could stop using it. If you've never pulled a large net on the bottom of an urban river, it's something that you might want to try. Maybe the amount of both anthropogenic and natural stuff that comes off the bottom, most of which is very hazardous and covered with sharp trachinid mussels. This is just 2019 alone, and Ray, are you here? Raise your hand. Ray. Ray's become our trawling guru here. He's got a 20-something foot piece of rebar that we managed to thread into the trawl. This is a bucket half full of what I assume is Corbicula Asian clam, and of course we managed to inexplicably catch a large common carp in our trawl too. But unfortunately the trawling has been quite successful, so it looks like we're stuck with the baggage associated with it. The eDNA, we're very fortunate to have strong molecular partners on this project. That's our co-authors Chris Reese and Meredith Bartron with the Fish and Wildlife Service in Lamar, Pennsylvania. So I'm not going to get into the lab side of things, that's really their domain, but in the field at each site we're taking 2 replicates. Each is composed of 2 liters of water that's filtered through a 1.5 micron glass fiber filter. We have a standard decontamination procedure using a bleach solution that we undertake before sampling at each site. We're taking field blanks at the first and last site each day, in which we're just filtering deionized water we bring out from the lab. That's just a check to make sure that we don't actually have contaminated gear going from one site to the next. And an additional fail-safe, we always work from downstream up, so furthest from any goby population, moving sequentially closer in case our decontamination ever was not perfectly effective. And just a quick snippet of the lab methods for the folks who are interested. We're doing single species qPCR. We're using the Nevers 2018 marker, and our field reps are run in octet with the qPCR reaction. So getting into results, all of my results slides are formatted the same way, so you can see the survey period is color-coded, and the size of the dot is scaled to the number of goby captured. And this is a really boring slide, and it was a really boring slide last year and the year before. Bottom line is that minnow trapping has been a complete bust for us, and I don't have time to go into the details, but in the Midwest it's worked very well to monitor goby populations. It has not been effective for us. We did do a proof of concept test in 2016, and our traps were in fact able to catch goby in high density goby areas, but we're catching a lot of other stuff. We have not caught goby in our routine surveys, so trapping has been discontinued as we look ahead to 2020. Seining, not much better. Admittedly, the canal is pretty lousy seining habitat. You don't have a lot of these nice, wadeable littoral areas that you'd like, but we're only catching goby at our positive control site, ranging from 0 to 5 fish per seine pole. So again, not a real effective method, at least in this habitat type, so we've discontinued seining going forward. Trawling, unfortunately, somewhat effective. You can see at our Sylvan Beach site, our captures, most surveys were getting fish, and as many as 230 fish per trawl pole. That's an average, that red dot, in August 2017. And a little more interesting, you can see we're starting to pick up goby in the trawl in 2018 and 19 at Rome, and in 2019 at Utica. But then, at second thought, is that really that interesting? I mean, we said we had an angler caught specimen just a couple kilometers up from our Utica site. That was in 2014. So if we're only starting to catch goby in 2018 and 19 at Rome and Utica, that's kind of odd. It suggests that maybe we don't have the whole story, and that brings up the question, can environmental DNA get us a more integrated picture of what's going on, and more so than the spatially limited traditional gear can do? And the answer is yes, or at least yes-ish, it looks like. So this is shown as number of lab reps positive. So remember, we took 2 field reps, they're run in octet at the lab, so 16 possible qPCR reps. So this is just a quick and dirty way to visualize it. So 16 out of 16, that's the strongest signature that you can get here. So work with me starting from the west, so you can see Sylvan Beach, strong positive signature during every single survey. Makes sense, we know goby are abundant there. But this is where it gets fun. Look at Rome. No hits for the first 3 surveys. Then we get 1 out of 16 hits, that's August 2017, June 2018, and then the DNA signature explodes August 2018. If you remember what the trawl did, no goby until August 2018. So this is pretty cool because it means the eDNA gave us about a 1-year warning that goby arrival was imminent. So you get these 1 out of 16 hits, the eDNA saying, hey, I think I'm getting something, and then all of a sudden the eDNA explodes at the same time that we actually catch goby in our nets. So that's pretty cool. Also, Utica, another really interesting story. You can see the DNA signature was strong right from the beginning of the project, but again, you'll recall, we didn't actually catch goby in our trawl until 2019. So finally, in fall of 2018, at this point, after all these strong eDNA positives and no fish captured, we got frustrated, took out the backpack shocker, and just bounced around in a couple kilometers immediately upstream and trying to figure out what are we missing? Where is this source of DNA coming from? And wouldn't you know it, we found a high-density goby population. This is our eDNA site. This is about 1 to 2 kilometers upstream at Lock 20. Very high-density goby population. So again, the eDNA gave us critical information that informed some targeted sampling, and then we were in a position to have a much better understanding of the distribution of goby. So, of course, I'm telling you how great eDNA is, and a lot of you are probably looking at me skeptically as you see this stuff over here. So let's talk about that. This was August 2017. I remember getting off the phone with Chris Reese at our molecular lab and covering my eyes and saying, holy shit, like this is really bad news, right? I mean, either you've got bad data or you've got goby in the Hudson or some other hard to explain scenario. So this is one of the challenges associated with eDNA. And I spoke about these results at length last year. I'm going to give you the short version today, but the bottom line is this. We don't believe this is contamination in our samples because A, we sample from downstream up, B, we had field blanks and a full suite of lab controls that all were negative, suggesting we didn't have field or lab-based contamination, nor do we have a history of that during any other survey. So then you start, and we don't, of course, believe that the goby invasion front was anywhere near there either. So then you start making up stories about boats or dredging or hydrologic events that, you know, maybe mobilize some DNA abnormally, but you don't know. And that's one of the challenges associated with this method. You're using the presence of DNA as a surrogate for the presence of the organism. And often they're synonymous. This is an example where it appears that they were not. A little bit less problematic. In spring 2019, these light blue dots, you'll see the 3 sites downstream from Utica, we had some hits. That's a lot more intuitive. We know we had very high flows during the spring. We did our best to get out in a moderate period, but velocities were still very elevated. This is probably just downstream, increased downstream transport of DNA from our Utica population. And interesting though, under base flow conditions in the summer, we did repeat our hits at the Frankfurt site, so that's probably real, and that suggests that goby are in fact probably encroaching on that Frankfurt site, and we may expect to see them shortly. So, trying to make sense of all this, I guess the first take-home message before we even talk about methods is that this goby expansion is inexplicably slow. I mean, we've seen in the Illinois River and the St. Lawrence River, just about any other large riverine system you can look at, we see expansion rates often of 20 to 30 kilometers per year, at least in a downstream direction. And since 2014, we're looking at, I don't know, maybe 3 kilometers. We don't quite have the resolution to know for sure, but it looks like less than 1 kilometer per year. So what is that about? You know, and I'd be happy to chat with folks later if you have any hypotheses. This is our best one. We assumed, and I think incorrectly perhaps, that when the angler caught specimen showed up around here in 2014, we assumed that that was the invasion front from Oneida Lake that had pressed all the way through. I'm not so sure that's the case. Looking at the data set that we've collected, we don't have DNA hits in Rome, you know, very weak starting in 2017, and of course we don't have captures until 2018. So this suggests that Utica could actually have been a separate introduction point ahead of the invasion front, and this is not unprecedented. We've seen in the Susquehanna River, a dead goby was found with a hook in its back, I think in 2013, and you know, knock on wood, it looks like there was no establishment, but I mean, there's definitely a history of bait bucket introductions, and it's entirely possible that that's what we've seen at Utica, and that could explain why it's taken goby a little bit longer to get their feet under them and continue expanding eastward. Again, just a guess, but it seems plausible. As far as methods go, eDNA seems to be a winner. It seems to be a more sensitive screening tool, and it's been really instrumental in helping us understand the distributions in the Utica and Rome area. And one point that's worth making, Rome is the high point in the canal. So once you have goby on the east side of Rome, all that water is flowing in the same direction to the east. And we were a little bit concerned that that Utica population might give off enough DNA that it might saturate essentially the whole study area downstream, and that doesn't appear to be the case. We're generally only detecting DNA maybe at most 10 to 20 kilometers downstream of a population, possibly less, probably less. So that's pretty powerful, because then when you get hits, you actually have some idea of what it means, rather than just that it could be anywhere, you know, in a 90-mile range. Our eDNA results, in general, are on point and very interpretable based on our knowledge of the invasion front. But the August 2017 results, I'd say, are a sobering reminder that if you're in a management position, I wouldn't want to be solely relying on these eDNA results trying to make management decisions. I think the eDNA is a great tool, but I think it's best paired with some level of the traditional gear still. Whether you rely on eDNA and then when you get results that need investigation, you pull out the traditional gear in a targeted form. You know, there's different sampling designs you could use, but I wouldn't completely retire the traditional gear because eDNA results can be scary sometimes. Finally, this is just a little snippet of data. It's ripe with variability that I'm not really showing you in the interest of time, but at our Utica site, this is our mean catch of tessellated darters in our summer trawls, from about 150 to about 5 in a 4-year period. Again, we have some other data that's not so conclusive, but this is, you know, probably not a good omen as we think about the, you know, native benthic fishes in this area. Finally, this probably won't work because fun stuff never does in presentations, but this is our aquarium at the office. This is in no way scientific or defensible, but we conducted quantitative electrofishing surveys in 20 trips to the Mohawk River this summer. The goal was to quantify the, I guess, the baseline level of all of our fish populations and try to get some sense as to, once goby do invade, be able to identify those changes. So one of the unexpected findings, many of our tributaries to the Mohawk had recently been colonized heavily by rainbow darters, which are not a native fish to the Mohawk River drainage, and had already displaced a lot of the native benthic fish. So in our aquarium at the office, I guess that you'd say this is, it's actually working. We are, I guess, recreationally playing out the next round of invasion dynamics where the round goby then are going to move into these rainbow darter habitats. And there's probably someone here who's saying, hey, that's a greenside darter. Yes, there is one in here. But the darters and the gobies were approximately the same size. That's the dominant male. The dark one here is coming out to play. But they were approximately the same size when we started this about 6-8 months ago, and you can see the gobies now have turned into, you know, pit bulls, and the darters are really exhibiting a sneaking behavior. Just you know, they're surviving, which we weren't even sure would be the case, but they're certainly not thriving with the gobies around. So you know, no surprise here. We know that gobies are really the ultimate competitor when it comes to small benthic fish, but they certainly lived up to their reputation. So, with that, just a couple concluding points. We have a manuscript in progress that we're hoping to submit to transactions probably in the spring, looking at both the performance of the different methods and the invasion progress through the state. We'll skip that one. We're going to try to use the quantity of DNA in the water to say something about the relative abundance of goby. So in other words, relating DNA copies per liter to our trawl catches. It's not going to be perfect by any means, but I think we may be in a position where we can determine a lot from a little as far as goby abundance. Finally, in 2020, we're going to be back at it, trawling, eDNA, and we piloted backpack shocking of the nearshore habitats this past fall, and that actually worked better than we expected. The literature says, you know, they don't have a swim bladder, it shouldn't be a great method, but we actually did pretty well with it. So I think we're going to replace the seining and trapping for the nearshore habitats and backpack shock instead. And finally, I want to acknowledge the Mohawk River Basin Program provided the majority of the funds for this project, and need to also acknowledge our molecular partners, Chris Reese and Meredith Bartram at the Northeast Fisheries Center, Fish and Wildlife Service. They've been really helpful for folks like us that are sketchy on our genetics. So, and with that, I'm happy to take any questions. Thank you. Karen?
Oh, the further exciting adventures of slowby.
Yes.
I was wondering if it's time to start looking in potential predator guts, like the bass.
I think so, both in terms of areas that are not colonized to get a baseline, and in terms of areas that are colonized to see how heavily they are, you know, in fact, relying on goby. I mean,
They're starting to learn to eat them there.
Yeah, I mean, it's no secret that smallmouth bass tend to have increased growth rate and condition when goby come in, and maybe I painted too negative of a picture in my beginning slide because there are positive effects of goby invasion, and yeah, I think you're right. I think it is time. Yeah. Right.
Maybe I mean, I think you could be right about Utica being a separate point of introduction, and maybe they just need to reach carrying capacity in Utica Harbor. I mean, that's a pretty big harbor area before they start spilling, downstream.
So it's a density dependent thing, and then the migrants go out eventually. Yeah, no, I think that's very believable for sure. Yeah. Great. Thanks, everyone.
Thank you.